So good afternoon, everyone. Obviously, I'm delighted to open and to chair this afternoon session of this very exciting meeting. And obviously, I would like to start by thanking again the Société Française du Cancer and all the organizers, and in particular, Danijela, for this very inspiring day. So in perfect continuity with the first sessions, and thanks to two distinguished speakers, we'll now address this afternoon two important topics in the field of cellular plasticity. First, the role of tumor microenvironment in cell migration and cancer invasion, and then the interplay between cellular plasticities and the so-called cancer stem cells, and their implications in key steps of the metastatic cascade. Thus, it is now my pleasure to introduce Danijela VIGNJEVIC. Danijela is a group leader at Curie since 2013. Before that, she did her PhD with Gary Borisi in Chicago on filopodia formation in migrating cells, followed by a postdoc with Daniel Louvar at Institut Curie on the role of these filopodia in cancer cell metastasis. Nowadays, as a group leader, Danijela is primarily interested in how epithelial cells interact with their microenvironment, both in gut homeostasis and during cancer invasion. And that is what I imagine she's going to tell us about today. So please do not forget to use the chat to ask your questions during the Danijela's talk. And having said that, Danijela, the floor is yours.
Working. So what we will, I can hear myself. That's okay? Okay. So it was a nice introduction from Stefano, actually, nice transition, because the cancer cells are not alone. So they are surrounded by different stromal cells, so from their microenvironment, and not only the blood vessels and lymphatics, but also immune cells, and particularly fibroblasts, the cancer-associated fibroblasts, or CAF, that are responsible for remodeling and producing and cross-linking, stiffening the extracellular matrix in which all cells are embedded. They have different functions in tumors, so they are shown to both promote the cancer cell survival and growth, but they can also feed cancer cells, modulate immune system, by both, they are shown in different contexts that they can be pro-inflammatory or decreasing the inflammatory reaction. As well, they can have two opposite roles in a tumor spreading, in some cases reported to be opposing tumor spreading and metastasis, and in many other instances, they're shown to promote the tumor invasion, either by directly influencing on the cancer cell spreading, by pooling the cancer cells or leading their invasion, or indirectly by remodeling the extracellular matrix, forming kind of roads that cancer cells will follow. So this is what I'm going to tell you about, this dual role of the fibroblast in cancer progression. And the first barrier that the cancer cells need to overcome in order to disseminate throughout the body, it's the basement membrane, so the green line here. It's the physical barrier. It's a specialized extracellular matrix that separates the tumor from the stroma. And it's made by joint interaction between the epithelial cells and fibroblast. So in order to ask the question whether fibroblasts help cancer cells to overcome this first barrier, we made the in vitro system where we can culture cancer cells on the top of the native basement membrane that we isolate from the mouse mesentery, which we strip of mesothelial cells. We end up only with the extracellular matrix, on which on one side we put cancer cells, and on the other side we put fibroblasts that are isolated either from tumors, from patients, tumors of colorectal cancer, and those will be CAF, or from the same patient as a control, we also isolate normal fibroblasts, and we call them NAFs. So what we showed in those essays, I will be short because this is really published, but what we end up showing is that the fibroblast would help in the majority of cases cancer cells to overcome the basement membrane and invade on the other side. And how they do this, we can look at it in live. So it takes the process about 6 days. And it looked like that the cancer cell, the fibroblasts are pulling on the basement membrane. You can see them on many histoscenes here and here and here, before the gaps in the basement membrane appear. That what cancer cells would use to explore by shooting the protrusions and eventually translocating on the other side. And as they are pulling on the basement membrane, the fibroblast would remodel it. So in control situation, we find that those fibers are uniformly organized, either seen by scanning EM or by IFM, where we can look at the roughness or topography of the basement membrane, and we can look at also the stiffness of those different areas. Now, with fibroblasts, they would reorganize it in a way to create bigger bundles that would be of a higher stiffness and surrounded by areas of the cleared fibers, so holes in the basement membrane, and those will be much softer areas. And overall, the stiffness of the basement membrane would decrease in the presence of fibroblast. So now this remodeling of the basement membrane was dependent on contractility of the fibroblast. This is their major characteristic. Usually they are much more contractile than the normal fibroblast. And inhibiting actomizing contractility, in this case using the blebistatin, prevented formation of those holes, remodeling of the basement membrane, and also stopped invasion of cancer cells. In contrast, when we use the inhibitors of proteases, I'm showing you just the MMPs, but we also look at other proteases, that did not stop the invasion of cancer cells in majority of patients and did not prevent remodeling of the basement membrane. So we can still see holes inside. So that would kind of lead the conclusion that the physical capacities of fibroblasts to contract and remodel would favor cancer cell invasion. Now, what's happening in the second step when the cancer cells are present into the extracellular matrix before they reach the blood vessels? And in this case, this matrix is, again, have a different organization. Now it's more composed of collagen 1 and other ECM proteins. And for that, we design a different in vitro model where we can grow the cancer cells as spheroids, as aggregates of cancer cells that are embedded in collagen 1 that you can see here in blue, containing, again, fibroblast cuffs or NUFs from patients. And again, the same thing, we would see that the fibroblast would stimulate invasion of cancer cells by remodeling the extracellular matrix in a contractility-dependent and proteases-independent manner. But importantly, what we found is that the fibroblasts, as they do this, they bundle collagen fibers and decorate it with a new matrix by depositing the fibronectin. Now, if the fibroblast cannot produce the fibronectin, if you deplete the fibronectin using scRNA, that stops the invasion of cancer cells. And similarly, if we inhibit the integrins beta-3 either by scRNA or with the drug silengitide, that prevented fibronectin deposition without affecting ability of cancer cells to remodel the collagen and prevented invasion of cancer cells. So if I put it all together, so this is what we are thinking is that in the homeostasis, the extracellular matrix, both on the basement membrane as well as in the extracellular matrix in the stroma, is uniformly organized of different kind of the fibers. And during the tumor progression, the fibroblast that would turn out in a more contractile cuff would remodel the ECM, create the bundles, stiffening, cross-link those filaments, and create those stiffer and more aligned areas of fibers that cancer cells would maybe use to explore and use as a road to escape the primary tumor. So what I show so far is that the progression of the tumor is not solely a result of the crosstalk between the tumor and the stroma via biochemical signals, the ligands and then the receptors, but that the physical microenvironment, like microarchitecture of the ECM or the stiffness, also plays a major role. But as the tumor is growing, it's growing against this viscoelastic boundary, which is a stroma, meaning our ECM and fibroblast. And that would generate, so it will oppose the tumor growth and generate the increase in the intratumoral pressure or solid stress. That could result, for example, in the collapse of blood vessels and development of hypoxic regions in the tumor. Now, if you look at the tumor, so this is a colorectal cancer, but this is true for other tumors as well, in particular, for example, for PDAC, that the cancer cells are surrounded with the huge amounts of fibroblasts and their ECM. And they're creating this boundary or capsule that would oppose the tumor growth. So here we ask whether the fibroblasts, in particular their contractile capacity, would play a role in shaping the tumor, forming these capsules around cancer cells and preventing tumor spreading. For that, we use a mouse model where we can spontaneously get tumors. So due to the mutation in the Notch and P53, these mice will get tumors in the intestine about 8 to 10 months later. And just to guide you, the cancer cell will have a green nuclei and green membranes, while the fibroblast will have red or pink membranes in this case. So what we see is that the stroma, so not only fibroblasts, but all the stroma would encapsulate the tumor and also penetrate inside of the tumor and form those intratumoral capsules by creating the nest around the cancer cells. And I'll give you a few other examples. Again, the stroma penetrating in the tumor, forming these intratumoral clusters, look like that those clusters of cancer cells are now invading into the stroma. And if we focus on one of the areas and look at it now in a 3D, we can see how those fibroblasts are circulating and bracing the tumors, reshaping and creating those small kind of lobes or nets inside of the tumor. Now, to ask the question whether the contractility of the fibroblast is important for formation of those intratumoral capsules, we decided to take those organoids or tumoroids from the mouse I just described and then graft them into the immunocompetent mice in which we can inhibit contractility or myosin 2 specifically in alpha SMA positive cell. For simplicity, I would say CAFs. That will be subcutaneous tumors in this case. To guide you, the cancer cell will have a green nuclei in this case. The wild-type stroma will have red membranes, and the myosin 2 knockout fibroblast will have green membranes. So what we see in control tumors, again, even in those grafted tumors, we see these intratumoral capsules made of fibroblasts and collagens. In other words, you can the tumors look differentiated, right? We have areas of cancer cells circulated by the stroma. However, in the tumors in which the fibroblasts lose their contractility, it looks more mixed or less differentiated in a way. The cancer cells and the fibroblasts are completely intermingled. And we can now quantify these areas of cancer cells that are devoid of fibroblast and ECM by using big scans of several millimeters of a tumor. So here is a control tumor. We can just use then second harmonic to isolate the areas that are only containing cancer cells without stroma. So this is a control, and I'm going to show you in a bit the knockout. You can appreciate how more mixed is or how those areas without stroma are much smaller, and quantifying in a way to say the same thing. So that would say that the myosin or the fibroblast contractility would be important for formation of those intratumoral capsules. Can we study this in vitro? So for that, we use the colorectal cancer that we propagate in mice as a PDX before putting them in the culture as a tumor explants. And then we'll see what's happened. Just to point out, we never use plastic. Everything what I'm going to show you is done on the soft gels. So the stiffness of the matrix will not influence the phenotype you are going to see. So what we see here is that both cancer cells and fibroblasts would separate, segregate naturally, like in the tumor in vivo. The fibroblast, as those elongated cells, would kind of create a barrier that would prevent the tumor cells to expand. Sometimes you can see them even pushing them backwards. But on the same dish, we can see another thing that eventually the fibroblasts, as contracting too much, they would jump on the top of the cancer cells and constrict the cancer cells, inducing the formation of those buds that are lift off from the dish. And giving you a few examples how we look at this now to be really quantitative, we try to standardize these assays by using microfabrication, where we can have the islets of cancer cells with defined size surrounded by cuffs very similar to what we find in mouse tumors or in human tumors. And then we can label the fibroblast and cancer cells in different colors. So fibroblast red, they surround the cancer cell clusters. Eventually, they jump on the top of them and then start deforming those clusters, lifting off. So to study the mechanical interaction between these two types, we performed a traction force microscopy. And that allowed us to see that the cancer cells exert very little forces at the substrate, and that the forces that the fibroblasts exert are fluctuating over time, but eventually they start accumulating at the border, at the interface between the cancer cells and fibroblasts. And now, due to the circular geometry of the system, we can now decompose those forces into the tangential. That will be the forces corresponding to the migration of the fibroblast around the clusters of cancer cells, or radial, and those will be the perpendicular forces. There could be pulling forces, like red ones, or compressing forces, the blue ones. And now, if we average those forces circumferentially over time, we can build the chemographs as a function of a distance. So the black line would represent the border between the cancer cells on the left and the fibroblast on the right. And what we see is that those forces, the radial forces, are accumulating over time. And you can see that they're all blue. They are compressive forces. So to quantify this, we can see that the compressive forces, first, they are positive as the fibroblasts approach cancer cells, and then they become negative or compressive as they compress the cancer cells. How they compress? So the fibroblasts, indeed, they are contractile. They have they express the high levels of phosphorylated, meaning active myosin, both in vitro and in vivo in the tumors. Again, all the fibroblasts surrounding the cancer cells, they express phosphorylated myosin. And now if we inhibit myosin using blebistatin, we immediately see a relaxation of fibroblasts. They don't contract anymore, and then relaxation of respreading of cancer cells. And which is very clear in those chemographs, that as soon as the drug is added, inhibit myosin, the forces drop to zero, and the cancer cells start expanding their areas. Okay, so now what will be the mechanical interaction between the two cell types? So for that, we use the laser ablation to disconnect the cancer cells from the fibroblast in red here, and then look at for the displacements, let's say cells in the area closer to the cut and further away from the cut. What we see further away from the cut is a control, very, very little displacements. However, closer to the cut, we see that the cancer cells are displaying, and if you look at the probability of angles, they are displacing toward the cut. In other words, that would mean that they were under compression in the tumors. What's happened with fibroblasts? So we can do the same experiment, but now cutting perpendicularly the fibroblast, and again, looking at the displacements in the areas closer to the cut or further away. And to simpler words, what we find is that, again, cancer cells are displacing toward the cut, meaning they are under compression, and the fibroblasts are recoiling in the opposite direction, and that would suggest that they are under tension. The same experiments now still in vivo. So those are the control tumors. We can inhibit myosin, and we see that both fibroblasts and cancer cells do not respond anymore. And if we take the explants of the tumors from the knockout myosin, of course, the fibroblasts are not under tension anymore because they don't have myosin, but also the cancer cells are not displacing toward the cut. And that would mean that in the tumors, the compression of the cancer cells is driven by contractility of fibroblasts. And the same thing, experiments we can perform in vitro in co-cultures, and we would see the same effect. Now, the question is, do cancer cells sense this compression, and can they respond to it? And for that, we look at for localization of mechanotransduction protein that Stefano introduced, YAP, that can shuffle in and out of the nucleus depending on the mechanical status of the cells. So in the freely growing islets of cancer cells, we see that the cancer cells, most of them, they have the YAP, which is in the nucleus. And we developed the method to quantify this using the correlation of the YAP signal with the DNA. So the more red signal that you see, that would suggest more nuclear YAP in those cells. Now, if we confine the cancer cells on the extracellular matrix patterns, we see that that itself induces partial exit of the YAP from the nucleus to the cytoplasm, but not to the degree when the fibroblasts are compressing the cancer cells. So if the fibroblasts are present in co-cultures, we see complete exit of the YAP from the nucleus to the cytoplasm. And that's not observed, or it's completely reverted, if the fibroblasts are able to confine the cancer cells, but they are not able to compress because they lost the myosin II. We see again, a relocalization of the YAP into the nucleus. So that would mean actually that in vitro, the cancer cells are able to feel the compression of the fibroblast and modulate the YAP localization. The same thing we see in tumors in vivo. So in the control situation, when the fibroblasts compress cancer cells, the YAP is mostly cytoplasmic. And in the situation when cancer cells, fibroblasts are not contractile, the YAP is now become more nuclear. Now, the next question was, can fibroblasts compress any material, or this is really induced by cancer cells? And for that, we generated with our collaborators giant pillars made of PAA. Again, it's soft gels. What you can see here are the beads that we can use to measure the forces. And around those giant pillars, we would plate the fibroblasts that would organize into these rings like around cancer cells containing actin and myosin. And now we can look through the section of this pillar and see whether the fibroblasts are able to deform the pillar. So in the green is a control situation. In red, what you can see is deformation of the pillar in the presence of fibroblasts. And Xavi Trepa and Ernest Emmanuel in his lab, they help us to analyze this more in 3D. So those are the real data just magnified 3 times in order to see how fibroblasts that are approximately where the red signal is are deforming the pillar. And this is the top view of the same experiment. Now, inhibiting myosin prevents deformation of those pillars. The same data, you can see it now if you unwrap the pillar, and you can see the compressive forces in control situation, inhibiting myosin, complete loss of compression by fibroblast. And the final question we ask is how the fibroblasts do this. Those pillars or cancer cell islets are giant, much bigger than a single fibroblast. So in order to compress, the fibroblast needs to be connected to each other. And one obvious point would be that they are connected with cadherins, like N-cadherin would be the first candidate. Unfortunately, depleting the N-cadherin did not affect the ability of fibroblasts to contract. So they were able to compress the pillar and also the cancer cells. So we turn out to the alternatives. Maybe the fibroblasts are connected indirectly through the extracellular matrix, collagens or fibronectin, because this is one of the most upregulated ECM proteins that we found in different patients. And I'm showing you the tumors in control situation. Wherever we see cuffs, we see fibronectin. If the tumors, they contain myosin knockout fibroblast, there is much less fibronectin present inside. So it's a good candidate. And in vitro, the same thing. Fibronectin is only produced by cuff, not cancer cells. And it becomes more linear or more organized at the interface. And in some cases, we cannot even distinguish those fibronectin fibers between individual cells. It looks like one giant cable that is circulating cancer cell lines. So if we silence fibronectin, the fibroblasts lost the ability to compress the pillar. They also lose ability to, reduce the traction forces in our co-cultures, as well as they lead to a partial entry of the YAP into the nucleus in the cancer cells, meaning that the cancer cells, in this case, are confined but not compressed if the fibronectin is depleted in fibroblasts. So that leads me to the model. What we are thinking is happening that the fibroblasts that surround the cancer cells, they can organize into these supracellular rings that are, those fibroblasts are compressive, and these forces are then reinforced through the production of extracellular matrix, fibronectin, maybe collagen as well, and that contributes to the supracellular contractility of fibroblasts and compression of cancer cells. And deformation of tumors. I will stop here because I would like to speculate whether this is good or bad for tumor progression. I think that this capsule of fibroblast around tumor would be a good because it would restrain tumor growth, it could restrain tumor spreading or maybe metastasis, but also could be bad because it would prevent infiltration of immune systems. And now if the fibroblasts are not contractile, now the fibroblast and cancer cells are completely mixed. Immune cells would have easier job to infiltrate these tumors, but also the cancer cell will have now a free escape route, so they could be able to disseminate. We're starting to look into this, and I'm gonna just show you where we are. We are building the same mouse model, but in this case, we are orthotopically implanting organoids into the colon mucosa and following the tumor growth by colonoscopy. And a few glimpses, no conclusion, just to show you where we are, that it looked like higher proliferation in those myosin knockout tumors. However, if you look at the big areas of the tumor, not specifically one single area, but really looking the whole tumor, the situation is a bit less clear. It looked like that there is a less proliferation in the myosin knockout tumors. An interesting thing is that, because I put those slides this morning to connect to the talks of a few speakers from today, especially the Thomas, we also look at the EMT program. And in the controlled tumors, when the tumors are differentiated and the capsule is formed, the tumors will have less nuclear ZEB1. And if the myosin is depleted in cuffs, that looked like that the translocation of ZEB1 into the nucleus in starting the EMT program. And it's really striking in vitro. So while the cancer cells spreading freely, they will have, in this case, it's snail and slug, would be nuclear. However, if they are compressed with cuffs, then at about 5-6 rows of cancer cells, they lose snail and slug into the nucleus, and meaning a reversion of the EMT program. And I'm going to stop here and thank the people who did the work. So the work is done by 2 postdocs, so Jorge Barbazan and Carlos Perez Gonzalez. And I would like to thank my collaborators in Barcelona, as well as people here at Curie. And thank you very much for your attention.
Thank you very much, Daniela, for this really beautiful talk. So now I would like to open the discussion. I see that we have several questions. Actually, I'm going to share a question with Fatima Meshta-Grigoriou. Considered as a whole, CAFs have several skills, so as you've shown, impacting tumor development. But so as shown by Fatima, and as you know, and some others, they are also highly heterogeneous. So my general question is, what do we know about the role of cell plasticity in the context of this heterogeneity and of this diversity of CAF subpopulations within the tumor? And a more precise question by Fatima, actually after saying that your talk is impressive and very nice. So she said that she would like you to comment the role of CAF expressing the myosin 2 and to which extent they are heterogeneous too.
Right. So this is a question that we're always having in mind. First, to tell that the fibroblasts that we are using are heterogeneous populations. So what I'm saying, CAFs are probably containing different kind of fibroblasts inside, and we don't distinguish them. Another thing that I would like to point out, that we now have the evidence that the fibroblasts even in the normal colonic tissue are very heterogeneous. We have the different populations of fibroblasts. Some of them are contractile without producing the ECM, and the other fibroblasts that are more ECM producing or growth factor cytokine producing, but not contractile. So what is the CAF of the origin in the tumors? I don't know. It could come from all of those subpopulations that are already present in the normal colon, or we will have a selection of some of those cluster or subpopulations that are overrepresented into the tumors. So that would be my view, that the fibroblasts indeed have different roles, and that maybe the subpopulation of fibroblasts, and they would exert different roles, but there is also attempting possibilities that the fibroblasts change their functions as the tumor progresses. So that maybe at the beginning they are playing a good guy and then encapsulating the tumor, forming this capsule, preventing tumor growth and so forth. By doing it so well, as I show in the beginning, it looks like that they are able to compress and even deformed tumors and detached to smaller clusters, what pathologists would call budding. And maybe those buds now will have higher chances to disseminate and spread throughout the body. So I don't have the answer. It could have been that different functions of fibroblasts would, some of them will promote and some will prevent tumor spreading, or it could have been that also fibroblasts change over time and from good guys to the bad guys.
Thank you, Danijela. Another question, I've just read it. Do you think that this kind of dialogue between tumor cells and stroma is only true for epithelial tumors? Any evidence in other types of tumors?
So what I just described about compression of fibroblasts to the other epithelial tumors, we don't have evidence that this is happening, but I would say yes. The crosstalk between fibroblasts and cancer cells is evident in many different types of tumors, ovarian, breast, as Fatima is working on, pancreatic tumors, and many, many, many others. However, this compression of fibroblasts by cancer cells by fibroblasts, we only demonstrated in colorectal cancer. I don't know if that was the question. Well, although I can say that the fibroblasts, what are called in the normal tissue or more myofibroblasts, would be able to shape the epithelium even in the normal situations during the development. For example, during the mammary, so branching morphogenesis in mammary gland or even in the lungs. So I would say generally the fibroblast would have these roles to compress and to shape the epithelium.
Okay. A question from Caroline Louis. Thank you for this fascinating presentation. It's not a question, it's an affirmation. A very naive question now. Does aging induces a defect in myosin expression in normal fibroblasts?
So this is the answer. I don't know, but listening to Stefano, I would say yes. And this is something that we are going to look at it. So that it looked like that this myosin knockout fibroblast would behave as aged fibroblast. And that could, that would be interesting to look whether what is the relationship between the aging fibroblasts, let's say in the gut, and those myosin KO in the young mice, whether they have the same phenotype. But it was very stimulating, this talk just before about that.
Two questions coming from Giorgio Seano from the Institute Curie. First, which is the order of magnitude of these in vitro CAF-mediated forces in terms of kilopascal? Wow, very specific and precise question. Do you think it is comparable to what happens in vitro? In vitral, sorry?
The answer is whether it's comparable, I don't know, and it will be very hard to know how much is in vivo. We do have some projects in the lab where we will be actually looking at the compressive forces in the tumors by different means, so in the real tumors, in the mouse, and then it will be easy. Then I will be able to answer to you. Right now, I don't have idea how much would be that in vivo, so I cannot compare.
But from a very specific question to a more general question, what do you expect is the consequence of these forces over the cancer cell resistance to therapy?
Whether the compression would affect the resistant therapy, we also have we are actually looking into this, so the student is following on the question. So it's not only whether the compression would influence the tumor growth and spreading, but we also have a project which I don't have the answer to tell you right now, whether it will affect the resistance to 5-FU and radiation or combination of the two. This is what we are looking right now.
Yeah. Question from someone you know, I believe, Daniel Louva. Which molecular mechanisms drive during fibroblast compression translocation of YAP from nucleus to cytoplasm? In other words, to transduce a mechanical signal into a change of cellular compartment?
So what is believed is that could be either a passive role, so just by pressing on the nuclear pores would be open and then the YAP would be YAP and other things could translocate in and out. That would be one way. Other ways could be the active transport of the YAP in and out. In this case, I don't know what it could be.
Question from Damien Grégoire. As different subpopulations of CAFs have been identified so far, do you think there are some specialized contractile CAFs coming back from our previous question, or this is a general feature?
Yes, so there are different subpopulations, both in normal gut tissue as well as in tumors. And some of them populations are highly contractile, so even more So contractile, and there are even group which is super contractile, almost looking like a muscle.
Question from Isabel van Sunningen. Thank you very much for this, very impressive. It's not a question, actually. I thought there was a question behind, but just congratulations. So it was very nice talk, Danijela, Isabel says. And so do I have other questions? Yeah, there is a new one. From some of your images and many colorectal tumor specimens, the invasive tumor cells organize as small glands, which suggests there is some kind of basal lamina. How does that fit with the first part of the work, with the CAF helping tumor cells to escape the basal lamina?
So in those more invasive areas, that would be already invasive, meaning the pathologist would say that the basement membrane is broken, although they would be still encapsulated with some of the basement membrane molecules. Whether the basement membrane is real complete structures like in normal tissue, I doubt. So, but if you use the stainings, for example, for specific lamining subunits, I'm sure we didn't do it, but I'm sure we will see it there. Doesn't mean that it's intact.
Okay, thanks a lot. Actually, I don't see any other questions. Oh, see, some other new ones. Coming from Rama from the Cancer Research Center of Lyon. Hi, guys. Thanks for this amazing talk. Naive question. Did you try to add a temporal dimension to your assay to decipher the impact of the matrix stiffness at the early stage of the metastasis and later on once the tumor cells are spread?
In these assays, no, but in the so student of mine actually addressed this by stiffening the matrix before the onset of invasion or after the onset of invasion. What they found is that by stiffening the extracellular matrix before the onset blocked invasion. However, stiffening of the matrix after already cancer cells start invading was promoting dissemination.
And how far do you think?
It's very important, actually.
And how far do you think the CAF-mediated forces are homogeneous along tumor progression? So we are coming back to this evolution of tumor development and progression.
What we know is that, at least in colorectal, and I'm sure that is true in other tumors, that more and more, let's say, contractile fibroblasts seen by one of the markers like SMA would accumulate in as the tumor progresses. The more invasive tumors will have more SMA-positive fibroblasts. But that doesn't mean necessarily more contractile. I want to be careful about this.
Yeah. And the last question, maybe another one, I don't know. So I don't know if it will be the last one. I have to find it again because actually it moved. So I'm lost now. It moved quite a lot. So I lost my previous questions. Okay, that's okay. And Rama and Daniel. So a question from Jean-Baptiste Lopez. What do you think about synergy of YAP exclusion when compressed with other oncogenes of colorectal cancer cells such as APC, KRAS, and so on in your model? Or what do you think it provokes in this cancer cell fate or evolution?
I can't answer because we are using the cancer cells are unchanged, and this translocation of YAP is more or less simultaneous. So I cannot think how that could affect the other oncogenes, but I have to think about it.
Okay. I'm sure that I lost a question somewhere, but anyway, I cannot find it again. So thank you very much, Danijela. It was really very exciting and really beautiful talk. Thanks a lot, Danijela.